It is 2 o'clock, so let's get started. And what we were talking about on, let me get that out of the way, on Wednesday, we were talking about hemoglobin, where we were talking about just starting to talk about like how hemoglobin basically works. So just a quick refresher on that. Hemoglobin is made out of 4 subunits, so it's a tetramer. And it has 4 oxygen binding sites, so 4 hemes. And it has 2 different forms. One if oxygen's bound and one if oxygen is not bound. And hemoglobin has this phenomenon called cooperative binding, where when one heme of hemoglobin binds oxygen, the other hemes want to bind oxygen more strongly. So one oxygen binds, the other sites want to bind oxygen more. When we're looking at a binding curve, a sigmoidal shape tells us if we have cooperative binding, positive cooperative binding. So myoglobin, if you notice, there's no S because there's like there's no cooperative binding for myoglobin. It's more or less like a straight line that just curves out. While in hemoglobin, we have this nice sigmoidal shape showing us that we do indeed have cooperative binding. So that's where we left off on Wednesday. And so if we want to look at binding data, all we need to do to determine if something has cooperative binding is we make a plot where we see fraction bound. Remember, this Y means the percentage of molecules bound with Lincoln. And on our x-axis, we have our ligand concentration. So I already made these charts for us. And if we were to compare graph A with graph B, which one do you think has cooperative ligand binding? So I actually have a poll for this. So just by looking at A and B, right, which one has cooperative binding? Remember, cooperative binding has that sigmoidal shape. Okay. So it looks like the majority of people have gotten in. So it is B, right? So if we look at B and you're just looking at what the curve looks like, it does have like that S shape, while A is more of a curve, just a straight curve. So cooperative binding shown by a binding plot where you have fraction saturation versus ligand amount. You're looking for an S, basically. Sigmoidal binding, that means we have cooperative binding. Any questions about that so far? Cooperative binding, what we're looking for on a chart, anything like that? If not, we'll move on to our next subject. Why does it make an S? The reason why it makes an S is that it's hard. The hardest oxygen to bind is the first oxygen. So it takes more ligand for it to bind. Once the first ligand binds, it's easier. So that's why it goes up faster, right? So that's why this curve goes up faster here. And then when you're out here, you're completely bound. So you can't bind any more ligand, so it flattens off. That's why it's a sigmoidal. Wouldn't it be bad if not a lot of O2 is binding? we will talk about that, why hemoglobin works like this. There's a very important reason why hemoglobin works like this, because hemoglobin not only wants to bind oxygen, it also wants to release oxygen. It would be super bad if your hemoglobin never let go of oxygen, because then all your cells would suffocate, and we don't want that to happen. So it has to be able to release it as well and not be able to bind it. All right. So when we talk about cooperative binding, there is an actual another plot whose whole purpose is to determine if there's cooperative binding. So first, before I get into that, we do have a different equation to use. So when we are looking at a protein that has multiple binding sites, our fractional saturation curve, or equation rather, changes slightly. Remember, we looked at this equation on Wednesday for myoglobin without any of these ends, right? So what these ends are, they're not the amount of ligand that can be bound. So they're not that. What these ends are is that they're called a Hill coefficient, all right? And what a Hill coefficient tells you is it tells you whether your protein has cooperativity or not. So what you do is you make this Hill plot, and you don't need to worry about the axes of the Hill plot, right? I'm not going to ask you to make a Hill plot. I just want you to know what a Hill plot is. And what a Hill plot is it tells you if your protein has cooperative binding or not. And so you make this plot, and you basically just measure slopes, right? And the slope that you get out is called a Hill coefficient. We need to know what value of Hill coefficient means what, right? So for myoglobin, the reason why there was no n there when we looked at that equation is because n was actually 1, right? And in like math, and if we raise something to the power of 1, we don't actually show the 1, right? So if we have a Hill coefficient of 1, that means our protein has no cooperativity for binding, which makes sense for myoglobin. Myoglobin has 1 binding site. So if I only have 1 binding site, I can't influence other binding sites. There's no other binding sites to influence. Hemoglobin has a value greater than 1. Hemoglobin is actually 3. So if you have a Hill coefficient greater than 1, you have positive cooperativity. And again, positive cooperativity is if you bind a ligand, you want to bind, in your other binding sites, you want to bind that ligand more. Now, if you have a Hill coefficient less than 1, you have what's called negative cooperativity. So that means if I bind one ligand, I don't want to bind other ligands. So binding a ligand actually lowers binding affinity for my other binding sites. And that's a Hill coefficient less than 1. And I think on your reading guides, I always get this question, is it possible to have a Hill coefficient of 0? And the answer is no. The only way you'll have a Hill coefficient of 0 is if you're looking at a ligand that does not bind that molecule. So n is 0 is impossible. So your Hill coefficient has to be greater than 0. Any questions about the idea of a Hill plot or a Hill coefficient? And you don't have to memorize hemoglobin's 3. You just have to know, what does greater than 1 mean, less than 1, or what does equal to 1 mean? Right? So if we don't have any questions on that, we can move on. So this question, we were actually going to do some math where we look at the fraction saturation of hemoglobin. Do you have a question? Can you explain why 0 is impossible again, please? So I can do it on this actually with this equation, right? If n is 0, raising something to the power of 0 is 1, right? If I raise anything to the power of 0, it's actually 1, not undefined. So if y02 equals, any number raised to the power of 0, that's 1. So let me get my eraser out. So what your y02 equation would be, it would be 1 divided by 1 plus 1. That means no matter what amount of ligand I have, I'm always going to be half of my binding sites will be bound, which doesn't make much sense. So if I have like a very small amount of ligand or an infinite amount of ligand, I'm always going to be bound 1/2. We can't do that. So a value of 0 doesn't exist. Just mathematically makes no sense when we look at our equation. So Hill coefficients can only be greater than 0. They can only be positive too. That's the reason why they can't be 0. If it's 0, you're looking at a phenomenon that doesn't exist. Wouldn't it be the same as raising it to the power 1? No, 2 raised to the power 1 is 2. 2 raised to the power of 0 is 1. Right? 2 raised to the power of 2 is 4. 2 raised to the power of -1 is, I forget, what is that, 1/2? Something like that. All right, so this question was just to get you comfortable using this equation. So we're not, I need to make up some time. So basically what you do is you just, it's basically what we did on Wednesday with hemoglobin, or sorry, not hemoglobin, myoglobin, where PO2, you just plug in your PO2, you plug in your N, so N is 3, and P50, I'll give you. And so we were just going to solve that 3 times. So I'm kind of just going to skip that as just to move on. So just I'll put the answers up to this one. Just make sure like you know what the variables are, right? You can look at a problem like that, and I'll give you this equation too. That's no problem. So you'll have this equation, right? Just make sure you know how to plug it in. That's basically what I wanted to show with this question. So we're just kind of gonna, instead of me sitting here for like 5 minutes while people calculate it out, we calculated it out for myoglobin already without the ends, and it's the same exercise. One thing I do want to touch on though, is we talked about this idea on Wednesday again, where we calculated myoglobin, which has a P50 of like, it's in like the single digits. It's very low, has a low P50. And we said, myoglobin is only 50% effective at delivering oxygen from the cell surface to the mitochondria. And we had a very long discussion about how I calculated those numbers, how to get that information for those numbers. So one of the thought experiments I want to do is, well, let's say myoglobin had the same P50 as hemoglobin instead. How well would this new hybrid myoglobin, or let's say, how well would hemoglobin transfer oxygen from the cell surface to the mitochondria? So I'm going to work through this out. So again, I'm going to draw a picture. At the cell surface, oxygen pressure is 10 torr. At the mitochondria, it's 1 torr, right? And let's say in our bodies, we have a mutant myoglobin that has the same P50 as hemoglobin. So at the cell surface, where pressure O2 is 10, fractional saturation is 10, so that's the pressure, divided by K. Remember, K, KD, or P50 all mean the same thing for this problem. So in our equation, it says K, and in the problem we say P50, they mean the same thing. So fractional saturation at the cell surface is 10 divided by 28 plus 10, which is 0.263. So remember from our discussion on Wednesday, this means that 26.3% of our mutant myoglobin would pick up oxygen from the cell surface. At the mitochondria, where pressure is 1 torr, our fractional saturation is 0.034, which means at the mitochondria, only 3.4% of our myoglobin still hang on to their oxygen. Doing the same calculation that we did on Wednesday, if 26.3% bind oxygen at the cell surface, that's this number, and at the mitochondria, only 3.4% are left with oxygen, that means with our mutant myoglobin, or basically with hemoglobin, we can only 23% of our molecules are able to deliver oxygen from the cell surface to mitochondria. Compare that to real myoglobin. So real myoglobin has a P50 of 2.8. And we calculated this out. 51.8% of myoglobin can deliver oxygen from the cell surface to the mitochondria. Hemoglobin is way worse at this. Hemoglobin can only do like 23%, which tells us hemoglobin has a different role in the body. So myoglobin's role is to help facilitate movement of oxygen from the blood to the mitochondria inside of a cell. Hemoglobin's role, hemoglobin can't do that well. Hemoglobin's kind of crap at that. What hemoglobin does, and what we're going to mainly talk about today in looking over hemoglobin will take oxygen from your lungs and deliver it to your cells. So that is what we're going to get into next, just doing a big deep dive at understanding how hemoglobin works. And really, we're going to be studying this because hemoglobin's so well studied as a whole, and just to get us comfortable with the idea of ligand binding to a protein and how proteins change upon ligand binding. So that's why we're going to learn so much about hemoglobin coming up here. But any questions there? I believe this is my last slide. Yeah. So any questions while I change slides here? So I want to go to the 7th. And that should say. All right, so let's get into our new PowerPoint. All right, so the first thing we're going to understand, or look at rather, is how does hemoglobin have this positive cooperativity? All right, and to do that, we have to understand the structure of hemoglobin, right? So here, this porphyrin ring, this makes up the heme. And myoglobin has two different states. Tense state, we also call that the T state. It's blue, that's no oxygen. Red state, relaxed, oxygen. So one thing you need to remember is that the tense state doesn't have oxygen, while if you're in the relaxed state, you're all good. You have plenty of oxygen. You are relaxed because you're breathing. You're relaxed. You have oxygen. While you're in the tense state, you're suffocating. You don't have oxygen, so you're tense, right? So remember that. Tense state versus relaxed state. Now, let's see what happens when oxygen binds. So let's say we're in the blue tense state. So no oxygen is bound. When oxygen binds, what happens is that the iron moves down a total of 0.6 angstroms. 1 angstrom is 0.1 nanometers. So it moves down. It doesn't sound like a lot, but for a protein, it moves down quite a bit. So this iron moves down to go and interact with the oxygen. When this iron moves down, remember we have a distal histidine that is non-covalently attached to the iron. So the histidine is going to follow this iron. So the histidine gets pulled down as well. When that histidine gets pulled down, it's connected to all of the other amino acids via peptide bonds, right? It's part of a protein. So if histidine gets pulled down, all of these other amino acids get pulled with it. And this barrel that we're seeing here, this barrel represents like all the amino acids. All of the amino acids shift because this one histidine gets shifted, and this one histidine gets shifted because iron is trying to go closer to oxygen. All right. So that's what happens in one subunit of hemoglobin when oxygen binds. Why does that matter? Why do we care about that? Well, remember, there are 4 different subunits of hemoglobin, or 4 different peptide chains that make up hemoglobin. So let's say the peptide, the subunit I just drew with an X, binds oxygen. This whole peptide will go through a conformational change. That is, the amino acids will move as shown here. And all the connections, and that's what we're showing in this picture. In these pictures, we're showing all of the amino acid connections shift. So here on the left, this is the T state. Here on the right, this is the R state, right? Before and after oxygen binding. So the way these different subunits talk to each other, the way the other hemes know that one subunit has bound oxygen is that they are interacting via amino acids. They have salt bridges. They have hydrogen bonds. They have hydrophobic interactions. When one subunit of hemoglobin shifts into the R state, all the other subunits are aware of this because different interactions change where the subunits meet. And this change of interaction is like a switch to the other subunits. The other subunits now know, oh, one molecule of oxygen has bound to us. We are now going to be more receptive to binding oxygen ourselves. And what happens is that their own ions start to be pushed down to be in a position to better bind oxygen. All right. So that was kind of a lengthy explanation. Was there any questions, or did I lose people on what's going on here? Like, what happens to the protein when we bind oxygen? Yes, R state binds oxygen better than T state because that iron is pulled down a little bit. That iron's in a better position to interact with oxygen. It's closer to oxygen. So yes, the R state wants to bind oxygen. The blue state does not. Or rather, red state, relaxed state has higher affinity for oxygen. Tense state has lower affinity for oxygen. That's more correct to say. Right, if there's not any more questions, we can move on. So this slide basically just has everything I said, just summarized, right? So hemoglobin, all 4 subunits are coupled. That is, you have a lot of interactions between them. And if you have tertiary changes in one subunit, that leads to a quaternary change in the whole structure. Now, hemoglobin is rather simple and only has 2 states, T and R, and it switches between those. T state, lower affinity for oxygen because iron's further away. If one subunit binds to oxygen, iron's pulled towards oxygen. Pulling up iron causes a bunch of conformational change in the alpha beta interface and at the C terminal interface. That's what the pictures were showing in the previous slide. When this happens, all the subunits can now adopt the R state. Now all the subunits want to bind oxygen. This is the molecular basis for positive cooperativity. This is how positive cooperativity can exist. And it's all due to electrostatics, all due to hydrophobic interactions, all due to amino acids, right? It's basically, physical biochemistry at that point. So that's our hemoglobin summary of what the last slide really says. So I do have a question that ties us back to Wednesday. So what we're seeing in this picture, it might be cut off. Here we have myoglobin. Here we have the beta subunit of hemoglobin. They look nearly identical. They look like almost the same. There's not much difference. If we compare the sequence of myoglobin to the alpha subunit of hemoglobin and the beta subunit of hemoglobin, there's like hardly any difference, right? There might be a difference, like there's a difference, but there's not that much structural difference between the 2 subunits. So let's say you had a mutation that replaced one of your hemoglobin subunits for myoglobin. Would this work? Why or why not? What exactly causes it to go from T to R? That whole conformational change I was talking about. Oxygen binds, an iron molecule is pulled closer to oxygen. That iron molecule is connected to a histidine. That histidine is connected to the rest of the subunit, so everything gets pulled down. That's a tertiary change. That one subunit is interacting with the other subunits via salt bridges, via hydrophobic interactions, via hydrogen bonds. Those are broken and new ones are formed. The formation of new interactions between subunits causes the other subunits to change, and they will adopt the R state as well. Change in tertiary structure leads to that overall change in quaternary structure. So that is what's going on. Hey, there we go. I think it could substitute, but you'll lose the positive cooperation because it's not used to linking with other stuff, more or less. So it could fit in there because they look exactly the same. The problem is myoglobin does not have the necessary amino acids it needs to properly interact with the other subunits of hemoglobin. So let's say that we had 4 subunits, right? We had a hemoglobin, 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 and myoglobin. This hemoglobin binds oxygen. Myoglobin down here would not have the necessary amino acids to understand what's going on with the hemoglobin that bound oxygen. It does not have the amino acids there to receive the signal. If you want to think about the other way too, myoglobin, if it binds oxygen, does not have the amino acids needed to interact with the other hemoglobins, right? So the other hemoglobins would not have a conformational change. It's all due, and most of biochemistry, when we talk about proteins, this is what's going on. You have this elaborate, very elaborate series of intermolecular interactions. Black does not work so well there. Intermolecular interactions. And based on the change of these intermolecular interactions, you have large shifts in structure, right? And this can change how things interact. Myoglobin does not have these amino acids, so no, it would not be able to substitute for hemoglobin. It could fit there, but it would have no positive cooperativity. Questions so far about any of the things we have touched on? All right. So here's just a question to test your understanding. I have a pool for this, but in a disease where you create hemoglobin Yakima, cysteine 99 is mutated to aspartic acid 99. This stabilizes the R state. Does the P50 go higher or lower? And let me get the poll up. Stop sharing those results. There we go. And in hemoglobin Kansas, asparagine 102 is mutated to threonine 102. This stabilizes the T state. Is the P50 higher or lower? And I simplified that question for you on the poll. I'm asking you, if you stabilize the R state rather, does your P50 increase or decrease? If I stabilize the T state, does P50 increase or decrease? So this will test your understanding of what does the R state do, what does the T state do. And this will also test your understanding of what does a P50 mean. Some interesting results so far. Looks like I need to go over some terminology again, which is okay. That's why we ask these questions, to see, where we're all at as a class and make sure we're following or not. Okay, I'll just end the poll here. Share those results. All right, so let's go over some terminology. The R state has a higher affinity for oxygen. The T state has a lower affinity for oxygen. All right. P50, the concentration of O2 needed to be 50% bound. That is the concentration of O2 that needs to be around for 50% of my binding sites to be bound. All right. So if I have a mutation that stabilizes the R state, that means I'm always in the R state, and my affinity for O2's increase, P50 is decreased, right? Oh, sorry, P50. Because it's easier to bind oxygen, so I'm going to be saturated, or I'm going to be bound with 50% oxygen at lower amounts because I want to bind oxygen more in the R state. While in the T state, I don't want to bind oxygen. So P50 increases. It takes more oxygen in the environment for me to be 50% bound, right? So don't forget what these terms mean, right? R state, greater affinity for oxygen. T state, lower affinity for oxygen. P50, the amount of oxygen that needs to be around to be 50% bound. So yeah, R state should have been lower, T state should have been higher. Questions about those ideas or our terminology here? All right, let's move on then. Okay, let's talk about the Bohr effect. The Bohr effect is an incredibly cool idea. The Bohr effect says, or what the Bohr effect is rather, and I know some of that's cut off at the bottom, I'll redraw it. The Bohr effect is under acidic conditions, hemoglobin binds oxygen weaker, right? So when you bind, so when you're more acidic, you bind oxygen weaker. And that's shown over here on this chart, right? So if I just draw a line for P50, right? At a pH of, sorry, that's not the best line, but at a pH of 7.6, my P50 is 20. At a pH of 7.4, which is more acidic, my P50 is like 28. Even more acidic, my P50 is like 33, right? So the lower the pH, the less I want to bind oxygen. Why does this matter? Why does hemoglobin have this ability? All right. So imagine that you are, I don't know, in a 5K fun run for some reason. You're one of those people who run for fun. I don't know why you think running is fun, but you do. So you're running, and your legs are, using a lot of ATP, and those tissues release CO2. So tissues that are actively, using ATP or respiring, they release CO2. Now, CO2 cannot be transported in the blood. CO2 is insoluble, right? So CO2 is insoluble. So what happens is that when CO2 gets exported out of the cell, there's an enzyme called carbonic anhydrase. And what carbonic anhydrase does is that it will combine water and CO2 and make carbonic acid, right? So it changes that CO2 into an acid, which means any muscle tissue that is doing exercise will have acidic blood. The pH will be lower around those tissues. Well, if there's hemoglobin around the muscles, what hemoglobin will do is that they will bind the acidic hydrogens, right? They bind those extra hydrogens. When it binds those hydrogens, the T state becomes stabilized. When the T state becomes stabilized, you don't want to bind oxygen, so you release oxygen. So I'm just going to repeat the beauty of that. Your muscles are exercising. They're doing work. The waste product is CO2. CO2 is changed into an acid. Your blood becomes acidic around your muscle. If hemoglobin's around, it will adopt the T state in the acid and release O2. That O2 is taken up by the muscle that is doing exercise. So this is how your body can deliver oxygen to tissues that are doing work. It's all because of the Bohr effect, and all because CO2 is a waste product. Your body is that refined to deliver oxygen to respiring tissues. All right. Before I move on with the rest of this plot, any questions about that? Just the idea of the Bohr effect of what's happening there with muscle. And if you're typing, I'm just going to rewrite some of this that is probably cut off at the bottom of your screen. HbO2, HbO2. All right. So let me just finish off with this plot. So our CO2 is changing to carbonic acid. This carbonic acid, when it reaches your lungs, another copy of carbonic anhydrase changes it to CO2. Now we can breathe out CO2 gas. The reason why we have HCO3 floating around, this is our blood buffer. Our cells don't work too well if the pH changes too much. One of the ways we keep our blood the same pH is by this mechanism. Yeah, so acidic conditions make it easier for hemoglobin to let go of oxygen for the muscles to use. Yep. Now, those of you who might hyperventilate, or have you ever been around a hyperventilator? What happens when you hyperventilate, right, is that you expel CO2 from your blood, right? Or not from your blood, from your lungs at a very fast rate. So if you hyperventilate, you are expelling CO2. And because of this equation, what will happen is that your blood will take carbonic acid out and keep making CO2 because of Le Chatelier's principle. Le Chatelier's principle is back from Gen Chem. If I unbalance one side of the equation, I'm going to move towards that equation. Basically, when you hyperventilate, you lose CO2, so your body makes more CO2 from your blood. What happens is that the pH of your blood goes up. So hyperventilation causes your blood pH to go up to like 7.6, 7.8, something like that. When you are that high pH, if we look on this graph, right, hemoglobin wants to be bound to oxygen. It does not want to let go of oxygen. So what happens when you hyperventilate, you make your blood more basic. Hemoglobin hangs on to its oxygen. Your brain doesn't get oxygen. You start to feel dizzy, and then you pass out. So all of this is connected, and we can explain all of this by just talking about hemoglobin, CO2, and oxygen. So that's what happens when you hyperventilate. To continue this chart, how this actually works with the body, right? In your lung, the O2 pressure is about 100 torr. So your lung is just full of oxygen. At 100 torr, all of your hemoglobin wants to bind oxygen. Then it'll go towards your cells where oxygen is lower or if there's acid. It releases oxygen. Myoglobin, that's what's shown here. Myoglobin picks up that oxygen from hemoglobin, brings it to the mitochondria. You release CO2, and that continues this path. What hemoglobin also does is that it'll pick up these acidic hydrogens, move those to the lungs so we can remake CO2. All right. So any questions about that wall of information that I just hit you with as I recover from being dizzy from hyperventilating myself? All right, if there's no questions concerning the Bohr effect, oh, looks like we do have one. Why do they give O2 gas for someone who's passed out? You pass out because your brain doesn't have oxygen. And so by flooding O2, I believe, inside your lungs and like your whole body, you're just again moving Le Chatelier's principle, where if like all hemoglobin are bound by oxygen, they're eventually going to want to give that up. So yeah, when you hyperventilate, you go into hypoxia, brain hypoxia. So you want to get oxygen to your brain. Also, I mean, when you're hyperventilating, you are expelling CO2. So if you can stop that, that's like why one of the things you want to do is breathe into a paper bag, right? You've seen that. Generally, that's making so you're rebreathing back your CO2, and then you're not changing your blood buffer too much. So when you get that O2 into your lungs, your hemoglobin will be like completely saturated, leaving your lungs, and then it can go to the other cell, can go to your brain, most importantly, and deliver that oxygen. And your blood buffer will go back to normal very fast once you stop doing that. Okay. But that is the Bohr effect. One last thing I want to talk about, and then we'll call a weekend and then do questions about this on Monday to see if you remember any of this from today, is the molecule BPG, right? So remember, well, let me go back to it. So I said, like, when we were looking at hemoglobin on, I don't have it anymore. When we were looking at hemoglobin on Monday, and there was this big center cavity, and somebody asked, you know, what's in this center cavity? And there's my internet. And I said, there's nothing in that center cavity. I actually lied. What's in that center cavity is a molecule called BPG, 2,3-bisphosphoglycerate. And what BPG does is that when it binds in that center cavity, it will favor the T state. So when BPG binds, it will force hemoglobin to be in the T state. While if you bind oxygen, so if I bind oxygen, BPG is expelled. Why is this good? Well, BPG in your blood allows hemoglobin to let go of that oxygen to your cell. So this graph in the top right, this line shows what hemoglobin behaves like without BPG. You can see P50 is really low. So if you don't have BPG, your blood does not want to get rid of the oxygen. While if you start adding BPG and carbon dioxide, your hemoglobin wants to give oxygen away. Now, this actually relates to why we get tired when we go to different heights. I'm struggling for the word, but let's say when you go to Denver and that's a mile above sea level and you can't breathe too well, that's all due to BPG, right? And that's shown down here. So at sea level, right, the pressure of oxygen is right here in your arteries, while at 4,500 meters, it's right here. That's your arteries, right? Your veins, it's the same no matter what. So when you go to high altitudes, that's the word I was looking for. When you go to high altitudes, let's say you fly to Denver tomorrow. Normally at sea level, 38% of your hemoglobin delivers oxygen from your lungs to your cells. So that's sea level. At higher altitudes, when you first go to a higher altitude, only 30% of your hemoglobin deliver oxygen from your lungs to your cells. That's why when you go to higher altitudes, you get tired so fast. You do a little bit of running, you're out of breath. However, what happens after a couple days at high altitude is that your body produces more BPG. So your body will naturally make more BPG. And what will happen, and it's shown on this red line, at high BPG in high altitudes, you can deliver 37% of oxygen from your hemoglobin, which is the same as at sea level. So that's why if you're climbing Everest, for example, what you do is you climb up a little bit, then climb back down, climb up a little bit and climb back down. Basically, you're allowing your body enough time to make BPG to get used to that altitude and lower oxygen. That's also why altitude training is good. So if you train at altitude where there's lower oxygen, your body makes more BPG to better deliver that oxygen. And so when you're at sea level where you have tons of oxygen, your body's better at delivering oxygen, you can run farther, right? So that's why if you're doing like long distance training, a good thing to do is go into the mountains, go into altitude to train because your body is better at delivering oxygen. Yeah, altitudes. Also, when fetal tissue has a different hemoglobin than adult tissue. It's actually called fetal hemoglobin. Fetal hemoglobin does not bind BPG well. Adult hemoglobin does. So the way that oxygen is delivered to the fetus inside the mother is that the mother's hemoglobin will get bound by BPG and release that oxygen. Fetal hemoglobin is basically unaffected or not affected as strongly. It can pick up that oxygen, and that's how the fetus gets oxygen from the mother's blood. It's all due to BPG. And all BPG is this tiny little molecule that we naturally produce, and it can bind in the center of your hemoglobins, and it will stabilize the T state. All right. Any questions about BPG and hemoglobin? And that will be my time for today, looks like. So if you are writing a question or thinking about a question, let me just go through my normal spiel. Next week, Friday, is our test. This PowerPoint is the last one that will be on our test. So when we move to a new PowerPoint on Monday, that won't be on the test. I will put homework up that will be due on Sunday as always. Other than that, if you don't have any other questions, that's all I have for you today. So thanks for stopping by. Please like, subscribe, and leave a comment. It's always appreciated. The PPG is different than one glycolysis, right? Yeah, so that's bisphosphoglycerate. No, it is the same thing. Sorry, I'm just looking at the name. Those are 100% the same thing. So yeah, the same thing from glycolysis is used here. 2,3-bisphosphoglycerate. So that's also a good lesson of the body uses these molecules in many different ways. So the BPG made in glycolysis is this BPG. All right. So if you have any questions, email me. Otherwise, I will see people later. Have a good weekend.